So again, thank you so much for the invitation. The title of my talk is From Surgery to Genomics. And what I'm going to describe to you is all our effort to dissect cellular and molecular heterogeneity in the most aggressive brain tumor in adult, which is glioblastoma. All right. So as you all probably know, glioblastoma is an extremely heterogeneous tumor. The standard of care of glioblastoma consists of debulking surgery where possible, and that is followed by concomitant radiotherapy and chemotherapy based on temozolomide. And this is then followed by a maintenance therapy based on chemotherapy, again, based on temozolomide. But unfortunately, despite all this surgery and treatment, the median overall survival for glioblastoma patients remains poor. It is only around 14.6 months, and the median progression-free survival is only 6.9 months. So glioblastoma is one of those tumors that contains many of the hallmarks of cancer. And one of the cardinal features is really that is extremely heterogeneous. And we believe that this heterogeneity is at the root of this poor prognosis for glioblastoma patients. Our work in this context of the heterogeneity of glioblastoma has been at the level of intra-tumor heterogeneity. So many people were focusing on inter-tumor heterogeneity, which means the differences observed in the biology of glioblastoma between patients. And there were several efforts trying to stratify patients and to develop personalized therapy. What it will suggest, and you can see on this schematic on the right, is that there are multiple molecular subtypes in glioblastoma, so patients can be stratified in different subtypes, such as mesenchymal, classical, proneural, and neural. What we did instead was looking inside the same glioblastoma at different areas of the same tumor. And this is summarized here. So initial studies were taking single samples from the tumor, and then they were profiling this single sample. And they were then assigning a molecular subtype to it. And in this example, you can see, for example, that you take a single sample from the mass of a glioblastoma patient, and then you profile it, and this is assigned by a lot of bioinformatic analysis and the integration of different genomic measurements to being a proneural subtype. Instead, what we did was to collect multiple samples from the same tumor in order to dissect intra-tumor heterogeneity. And what we found by doing that is that now there is a coexistence of the different molecular subtypes in the same tumor. So as you can see in this example, when we collect 5 samples from the same tumor, now you see that some of them are assigned to be mesenchymal and some other are assigned to be proneural. And so this is a problem in the sense that it suggests that it's very tricky to now stratify patients and develop personalized therapy based on this molecular subtype because the molecular subtype coexists in the same patient. And so we did work in this context, and other investigators also extended this work by adopting a better way, that is to look at single cell level. So we were looking more at tissue level by performing multiple sampling, and other people were looking at single cells, and they observed exactly the same, the coexistence of multiple subtypes in the same tumor. And so we believe that this extent of heterogeneity is really at the root of why glioblastoma is so complicated to treat, and still is also at the root of the fact that we probably have a very limited understanding on the biology and how this heterogeneity is developed. So in order to really dissect the intratumor heterogeneity, we need an objective method to perform this multiple sampling. And what we did during my postdoctoral training in Cambridge was to adapt a surgical tool which is based on 5-aminolevulinic acid, which is abbreviated as 5-LA, to then perform an objective sampling of these tumors. So what is 5-LA? 5-LA is a precursor of the M biosynthesis pathway, and this is usually administered before surgery because it allows a better resection. So what happens is that 5-LA, through a number of steps, gets converted in protoporphyrin 9, and protoporphyrin 9 has fluorescing properties, such as during surgery, if you adopt a blue light, you can see now the tumor becoming pink. And this is a video, thanks to Dr. Chon, that shows you exactly this. So this is a glioblastoma of a patient who got administered with 5-LA. And so if you again use a fluorescence microscope in the operating room, you now start seeing that all the tumor tissue becomes pink, and that can guide the neurosurgeon to perform a better resection. So this has been utilized because it allows a better debulking of the tumor. And we thought that this could be also utilized for our research purposes because it can also guide our multiple sampling analysis. And so in initial effort, we adopted this method. And, you know, as a starting point, what we did was to sample different areas of the tumor. So patients were administered with 5-LA, and we thought of really doing a proof of concept study in which we sampled the 5-LA positive material in the tumor mass, as you can see here in this schematic on the bottom left. And we also collected as a comparison the necrotic material from the same tumor, which by definition, because it contains necrotic tissue, is non-5-LA positive. And then we collected the margin area, which represent the interface with the normal brain. And again, 5-LA as this is really a powerful tool because it allows you now to better see the margin, to better see that interface, in the sense that that is the area in which the accumulation of 5-LA is much weaker. And so now you can objectively identify the margin in this glioblastoma patient. So we did that, and we wanted to understand whether now we had the opportunity with 5-LA to interrogate and to dissect heterogeneity in these three areas. And so as you can see, of course, to validate this method for research purposes, we had to look at the tissue that we were sampling. And so the tumor mass tissue, the 5-LA positive tissue, really was representative of all the glioblastoma histopathological feature, as you can see summarized in this table in the bottom right. So at the tissue level, we could see the majority of tumor cells in this area is expected, as well as histopathological feature of glioblastoma, such as microvascular proliferation, small area of necrosis, and we also performed the Ki-67 staining. It all looked exactly like what we expected from a tumor mass of a glioblastoma patient. When we looked at the necrotic material, as expected, we noticed that that was at tissue level completely different from the tumor mass, and the majority of the cells in this area were inflammatory. And then, more interestingly, when we looked at what we sampled as the margin 5-LA negative area, we observed that this looked exactly like normal tissue. And as you can see in this little image, the arrow point to what looks instead as tumor cells. So in general, it looked exactly like a normal brain tissue, but there were very few tumor cells that pathologists could spot when they were analyzing these slides for us. And so what they found is that mostly there were normal cells, but approximately they could see 10% of the tumor cells in this area. And so this suggested that, again, our sampling was correct, and that we, for 5-LA, we had the opportunity to interrogate the margin of the tumor and compare it with the tumor mass. So what we did next was to refine this method, and what we developed is what we called the Fluorescence-guided multiple sampling scheme, also known as FGMS. So this is a method, like I said, based on 5-LA, in which now we have that opportunity to sample the tumor mass, the tumor margin, and also to start characterizing at cellular and molecular level the heterogeneity of all this area. So in order to characterize the heterogeneity of the tumor mass, what we did was to collect multiple samples from the tumor mass. So in that initial pilot study that I told you before, we were doing just a single sample collection from the tumor mass. Now what we did in the following up studies was to collect multiple areas, and we were annotating where we were collecting sample in the tumor mass. And so here what you see is images by MRI in which we annotate the collection of these T1, T2, T3, T4 masses, basically sample from the tumor, the original tumor mass of glioblastoma patient. And so now we could, by only focusing on the tumor mass, look at the heterogeneity and having multiple samples from this area. And so we collected all these in a number of glioblastoma patients, and we published the work on the initial 11 glioblastoma patients on which we did this multiple sampling, and we applied the FGMS scheme. And this is what we did in order to summarize our finding. We were able to perform a genomic analysis on each of the samples collected for each patient, and also to perform a cellular functional analysis on each of the samples. What we found, which is interesting, I believe, is the fact that not only we could detect extensive genomic and phenotypic heterogeneity, but we could also use that information to understand the tumor evolution in each patient. So we used the genomic analysis on each sample, which we collected. In this case, is the tumor T1, T2, T3, and T4. So these are 5 areas collected from the 5LA positive tumor mass of this patient that is labeled as SP42. And when we did that, we genomically profiled each of them. We analyzed, we performed a lot of bioinformatic analysis, and we were looking at, again, the differences, the heterogeneity of the genetic alteration in each of these areas. And we could infer, by looking at the distribution of this genetic alteration, at how the tumor evolved. In particular, we could infer of the genomic profile of a putative founding clone for this patient. And so as you can see here, we could identify the potential driving alteration in the founding clone, and also we could approximately position where the founding clone was. And so by doing this approach, not only we detect an extensive intra-tumor heterogeneity, but we use that now to perform what is called a phylogenomic analysis. And so we could infer that based on this alteration and the putative founding clone, there was a divergence in terms of further genomic alteration, and that gave rise to additional clone and additional cells that then gave rise to the different area that we were sampling, T1, T2, T3, and T4. We were happy to see that this method, FGMS, has been also adopted by several other investigators for different type of analysis. So like I said, we did genomic and phenotypic analysis, and we were looking at tumor evolution. Other people have also done that, and they have used FGMS, and they were also looking, like us, at the margin of the tumor, and they were looking at tumor cell phenotype. So we were happy to see that this turned out to be useful for the interrogation of intra-tumor heterogeneity in glioblastoma patients. So now what I want to do is to summarize what we published so far. And then for the rest part of this talk, I'm going to present to you some unpublished work of the lab, so that you will see where we are going next in order to really understand better and to dissect intra-tumor heterogeneity in glioblastoma. So far, I've told you that by 5LA and by the FGMS scheme, we are able to dissect areas in the tumor. And we focused on dissecting the tumor mass by performing multiple sampling. And also we identified and characterized the infiltrative margin. But this method also turned out to be very interesting and very important for our study, because we were able to observe that there was fluorescence, so that fluorescence suggested the presence of tumor cells, as well, not only in the tumor mass, but also in the subventricular zone of many glioblastoma patients. And as you probably know, the subventricular zone is one of the most well-characterized neurogenic area in the brain. And so it was interesting to us to see that probably there were tumor cells also in this area. Because of the presence of 5LA also in the subventricular zone, we had the ethical justification to sample this area, and we went ahead, and for those patients in which we could see fluorescence in the subventricular zone, we also sampled that area. So we were performing multiple sampling on the tumor mass, on the infiltrative margin, and also on the subventricular zone. We performed genomic analysis, phenotypic analysis of all the cells isolated from this area. And what we found is that really this infiltrative margin and the subventricular zone are areas that are really not studied, not much characterized, And because now we had this tool based on FGMS, we can use to interrogate what we consider is a residual disease in glioblastoma. And residual disease is the disease that is left behind. And we believe that especially the infiltrative margin and the subventricular zone represent that type of disease. And so we went ahead and characterized both areas. And in particular, what we found by genomic analysis is that we were able now by phylogenomic reconstruction, so this tumor evolution approach that I was telling you before, to position as well the infiltrative margin and the subventricular zone in what we called our phylogenetic tree. So again, looking at the genetic alteration, not only we could infer the founding clone for each patient and the genetic profile of that founding clone, but we were also able now to look at the genetic alteration that were present in the infiltrative margin and in the subventricular zone. And by also performing longitudinal analysis, so we were comparing patients which underwent surgery for the primary tumor, then were treated and underwent a second surgery after treatment. We collected different, We collected different area, and we collected also the subventricular zone and infiltrative margin from this patient. And we were able now by genomic profile to again understand what is the role of the subventricular zone and the margin in glioblastoma patient. We were able to position those areas using tumor evolution approaches, and we were able to show that both areas contribute to seed the recurrent tumor. So all this is published, is a lot of genomic and bioinformatic analysis. So it's not super easy to understand, but I think collectively our findings showed that it's really important to characterize better these two areas and that they play a role in glioblastoma growth. So really focusing on this longitudinal, also called the spatio-temporal analysis of a glioblastoma patient, what we found is there something that we believe is very interesting. So by looking at two patients longitudinally, again, we sampled the original tumor and we sampled the recurrent tumor after treatment, we were able to see that the subventricular zone and the margin play a role really in seeding the recurrent tumor. So what we think is happening is that the infiltrative cells that are present in the subventricular zone and in the margin are important because when there is a resection of the tumor, which is summarized in this schematic, you have the tumor mass and the filtration in the brain, and the presence of tumor cells in the subventricular zone, which here is shown in blue, whereas the one in the margin are shown in green. Now you resect the one that are in the tumor mass, but unfortunately, the one in blue and in green, so respectively the subventricular zone cells and the infiltrative margin are left behind, and those give rise to the recurrent tumor. So this suggests that those infiltrative cells are really key, then that infiltrative process happens very early during tumor growth. So it's not that proliferation happens first only. There is proliferation of the tumor, but there is also a lot of infiltration. And that infiltration is responsible for seeding the recurrent tumor. Now, going back at this analysis of the infiltrative margin and the subventricular zone, what was very interesting for us was to observe that those two areas were different in terms of profiling. So gene expression profiling of those areas across several patients suggested that they represent different type of niches in the tumor. So here what you see on the left is a clustering analysis in which we were classifying patients, and from each of them, we were looking at the gene expression profile for the tumor mass, the subventricular zone, and the margin. And we were then analyzing those areas and compare them with this molecular subtype classification that I described earlier in proneural, neural, classical, and mesenchymal. And so what we found is that while 100% of the infiltrative margin sample always classify as the subtype labeled as neural, which is the closest to the normal brain, the subventricular zone sample, the majority of them, not all of them, but the vast majority of them, 77%, classify as mesenchymal, which is one of those subtypes that is characterized by the worst prognosis. Now, interestingly, work by the Cancer Genome Atlas showed that the mesenchymal subtype is that subtype that is characterized also by low tumor purity and the presence of cells that there is a majority of non-tumor cells present in there. In particular, there is an abundance of macrophages. So this suggested that we were able now by gene expression profiling to identify differences beyond what has been detected in the tumor mass, beyond what is known in the literature. Most of the work is done on the tumor mass. So we were able now to look at these two niches and trying to compare those niches versus what is known in the literature on glioblastoma. So up to this point, what I told you is that we were doing a lot of cellular and molecular analysis to look at the intra-tumor heterogeneity of this area, which are summarized here on the left. So these, what we call these glioblastoma niches, the tumor mass, the subventricular zone, and the margin. We use the 5LA, and as you can see, the tumor mass and the subventricular zone are 5LA positive, the margin is 5LA negative, but we were really focusing on the tumor cells that are present in this area. Right? So our effort was really towards the characterization of tumor cells in this area. But what is missing is the analysis of the entire microenvironment in each of these areas. And so now really this represents the next step for us, represent the foundation of my lab. So we are using now this same method, we have further refined it to better characterize each of the area. And so what I told you so far has been all published work. And if you have any question on it, I can also direct you on our publication. But now I'm going to present to you unpublished work, and that is currently ongoing in my lab. So again, we have FGMS implemented here at the University of New Mexico. We also have a collaboration with Dr. Chohan and Dr. Perkins at the University of Mississippi Medical Center. And what we do is to collect multiple samples from each of the glioblastoma patients that undergo surgery here at the institution. Like I told you, we have tumor mass as a comparison. We investigated infiltrative margin. We, where possible, collect the subventricular zone. So from each of these areas, what we do, we dissociate to single cells. But now we don't only select and enrich for the tumor cells, we look at the entire microenvironment. And what we do is to perform a single cell, single nucleus RNA sequencing, because that's the best resolution to allow us to interrogate each tumor microenvironment in these niches. But that's not sufficient, we believe, because it's very observational. It can give us a snapshot of what is happening in each of these areas, but we also need to perform what is called functional phenotyping analysis. So we perform a lot of functional assay on each of these areas and on all the cell population that we select from each of them. Now, the strategy really allows that we go straight from the operating room to the laboratory. We collect each area and we perform tissue dissociation. We have method, we have optimized method in order to allow the preservation of the entire cell composition in each of these niches. And in particular, we don't perform any enzymatic digestion. We don't enrich for selective cell population by doing cell sorting, and we don't put the cells in culture. So it's going straight from the OR to the lab and to this genomic and phenotypic analysis, because we don't want to alter in any way the cellular composition. We don't want to use any sort of selection. We really want to preserve the entire tumor microenvironment in each of these niches. So far, what we did was to collect at our institution 22 glioblastoma cases. And here now I'm going to present to you very briefly some of the preliminary data we got on the comparison of the tumor mass and the margin from these patients. In total, we have collected 72 tumor masses and 26 margins. So we believe we have reached, a good number on which we can perform a single cell, single nucleus RNA-seq and functional phenotyping analysis. So let me show you these unpublished data, which I think are very interesting. So we were looking at our first patient, GBM1. We were looking at the infiltrative margin. What we did here was to perform a single cell RNA-seq. Now, for those of you that are not familiar with this technique, just very briefly, what this technique allows is the ability to look at each single cells, all right, in that sample that you are analyzing. So each of the dots that you see here on this UMAP plot is a single cell. So on average, when you do single cell, single nucleus RNA-seq, you can analyze the genomic profile of cells, approximately 10,000 cells, and you're probably able to see 2,000 genes per cells. And in particular, what you look at by RNA-seq is the transcriptome of each of the cells, okay? So in an initial analysis, what we did was to infer, based on the transcriptome of each cells, as of why we had, you know, a different composition of cells inside that area. So we were looking at the amount of tumor cells versus the amount of non-tumor cells. And you may remember that at the beginning of this presentation, I told you that the margin is characterized by approximately only 10% of tumor cells. So we were happy to see that by doing now single cell RNA-seq analysis, we could infer that the majority of the cells in this area are actually non-tumor cells. So the cells labeled in blue here are all non-tumor cells. And as you can appreciate, they are the vast majority. Only a small amount are tumor cells, and are those in red. So now when we look at the transcriptome and we perform what is called the cell type annotation, so we try to infer what type of cells each of these is, we found that the majority of the non-tumor cells, right, the blue one, are assigned as macrophages. So this means that the infiltrative margin has an abundance of this non-tumor cell population, macrophages. And then what we did was to go ahead and look at the expression in each of these cells of specific macrophages marker. And so here what I'm presenting to you is the analysis, the summary of the analysis that we did. We looked at more of 400 markers for this cell population. And here I'm showing some of these markers to you. And what we did was to look at the myeloid panel. And as you can see, there is a very heterogeneous distribution of this marker. What you see here on the bottom on sort of the identity is basically by cluster. So this means that here on the left, you have a representation of each of the cells. But then when we do cell type annotation and we assign the cell to a specific cell type like macrophages or astrocytes, for example, we perform a cluster analysis. So different clusters are identified in this sample, and these are the cluster 1 to 15 that you find here plotted as identity, okay? So here we were looking at the expression of this marker in each of these clusters. We went ahead and we looked specifically at the expression of marker associated with macroglia and monocytes-derived macrophages. And what we found, as you can see by the expression of this marker, is that the majority of non-tumor cells in the infiltrative margin are monocytes-derived macrophages. And now what you know probably is that macrophages have two different activation states, M1 and M2. So we look specifically at marker associated with the M1 and the M2. And as you know, this activation status that is called M1 and M2 is very important because it can tell you of the functional identity of macrophages. We also looked at the corresponding tumor mass from this same patient. So we were looking, at performing the same exact analysis, trying to understand what were the difference at the level of tumor microenvironment between tumor mass and infiltrative margin in the same patient. And so the first thing we did, like I said, is to infer about the presence of tumor versus non-tumor cells in this sample. And again, we were happy to see that in the tumor mass instead, now you see that the majority of the cells, as expected, are tumor cells, and the minority are non-tumor. So opposite to the infiltrative margin. And that's expected, right? Because in the tumor mass, you expect a lot of tumor cell presence. We then performed again cell type annotation, and what we found is that now the majority of non-tumor cells in the tumor mass of this patient classify as macrophages. And we looked again at all more than 400 markers for all these cell population, and we performed exactly the same analysis. But going back to the infiltrative margin, which is the most important area for us to characterize, we then went ahead based on those data and we performed all cluster pathway analysis. So by looking at the transcriptome of each of the cells in this area, and in particular by looking at all these cluster assignment that we got by performing bioinformatic analysis, what we found, and is summarized here, is that there are different pathways that are active in population specifically associated with monocytes-derived macrophages. So if you look at this pathway analysis, what you see is that these monocytes-derived macrophages express genes that are associated with different biological processes. But the one that stand out are antigen processing, presentation, and oxidative phosphorylation. So that was interesting to us because it suggested that there is heterogeneity in this area. So while some macrophages can rely on specific biological processes, others may not in that same area. And so this was done at the level of all clusters, so a comparison between all clusters together. But then we went ahead and what we did was to perform a specific cluster-to-cluster analysis. So what this means is that we were looking at comparing monocytes-derived macrophages that were relying, for example, on specific biological processes such as antigen processing and presentation, and compare those versus those monocytes-derived macrophages which instead relied on other biological processes such as oxidative phosphorylation. What we found is that that type of analysis, cluster-to-cluster enrichment analysis, suggested that in fact, for those that rely on oxidative phosphorylation, the pathway that are most activated are mitochondrial respirosome and respiratory chain complex. So suggesting really that those macrophages that rely on oxidative phosphorylation are active in those biological processes. So this is very interesting because it suggests a functional heterogeneity of this cell population in the infiltrative margin. And there is a lot to do at this point because we need to go functional and characterize and really see whether the monocytes-derived macrophages in the infiltrative margin are relying on these biological processes by performing functional assays. All this is based on bioinformatic analysis and based on transcriptomic data. So to summarize where we are now, I hope that I showed you that by FGMS, we can perform a specific and objective identification of glioblastoma niches, and that we can preserve the cellular composition of the tumor microenvironment in each of these niches by having optimized the protocol in which we don't have any in vitro manipulation. So we are able to refine FGMS in order now not only to characterize the heterogeneity of the tumor cells, but now to look at the tumor microenvironment in all areas, in the subventricular zone, in the infiltrative margin, and in the tumor mass. We have performed the single cell, single nucleus RNA sequencing, and what this analysis so far has shown is that the majority of non-tumor cells in the infiltrative margin are monocytes-derived macrophages. But then when we looked specifically at all these markers, more than 400, and more specifically, we looked at M1 and M2 activation states associated marker, we found that those are not abundantly expressed in the margin, but also we found that they were probably really too basic in terms of characterizing the functional identity of macrophages in this niche. So what really we need to do is to use a more comprehensive analysis. And we started with pathway analysis, but definitely we need to go further and go more functional to better understand the identity of these macrophages. But up to now, this pathway analysis has shown that at least some monocytes-derived macrophages in the infiltrative margin express gene associated with antigen processing and presentation, and others instead express gene associated with oxidative phosphorylation. And so again, this points out to the fact that there is an extensive functional heterogeneity of this population. So, to summarize all this, I would say that initially our focus was really the cellular and molecular heterogeneity of the tumor cells. Now, this focus is moving towards a more comprehensive analysis of the cellular and molecular heterogeneity of the entire microenvironment. And because many of the cells in this microenvironment, especially in the infiltrative margin, are monocytes-derived macrophages, we are going to perform functional assays on this population. So I'll stop here, but before doing that and taking questions, I want to acknowledge all the people who did this work. And so this unpublished work that I presented towards the end of this presentation started with this very talented undergraduate student in my lab, Vanessa Avalos. She's now at Emory University and she has started a PhD. And is continuing now thanks to a very talented postdoc in my lab. Her name is Jamilette Vicon-Munoz. Here in the picture and listed are the other people of my lab that has been named the Brain Tumor Translational Lab. Of course, all this work is possible thanks to the generous donation of patient sample. So we are very grateful to all the patient and their family for donating sample and for allowing us to do this research. Here at UNM, we have a strong collaboration with facilities of the UNM Cancer Center, as well as a strong collaboration with the neurosurgery department. We have neurosurgeon working with us, Christian Bowers, Mike Kogan, and we have as well a research team that allow us to do this research. In terms of the resources that we have here in the Cancer Center of UNM, we have strong collaboration with the Analytical and Translational Genomics Center, as well as the Bioinformatics Center and the Human Tissue Repository, which are headed by Scott Ness, Yang Wu, and Dennis McCance. And so all this genomic analysis, transcriptomic analysis is done through a collaboration with them. But we also have a strong collaboration with Dr. Choan at the UMMC. And I'm very pleased that soon we will have as well data from our collaboration. And I also want to thank not only Dr. Choan for this constant support and collaboration, but as well Dr. Eddie Perkins, who is helping as well at the UMMC. So it's really an interinstitutional effort at this point, and I really hope that we can move forward and further develop a project based on this collaboration. And I want to conclude by also thanking all the funding bodies who are supporting this research. In particular, some of the work that is presented in this slide has been obtained thanks to the support of the Ben and Catherine Ivy Foundation, but also from internal support here at UNM. So with that, I stop here, and I'm thankful for your attention and participation, and I look forward to your question.
Thank you. Thank you so much for an amazing presentation. So we are open for questions. Dr. Chauhan, you want to comment before taking questions?
Fantastic, Dr. Piccirillo. As always, this is great work. I had a question about your macrophage analysis.
Mm-hmm.
What do you think these cells are doing there? Are they, what do you think about their role? Are they beneficial? Are they trying to get rid of the tumor? And a follow-up question, which I'm sure you have been asked before, is the use or the almost universal use of steroids in patients with, clinically, in patients with glioblastoma. As you know, every single one of our patients is put on steroids, at least perioperatively and postoperatively, sometimes even through their 6-week course of radiation, and then often on and off, depending on how they're doing clinically. So I just want to hear your thoughts. What do you think these macrophages are doing there? Are they good or bad? And what are the clinical implications if they are good?
Mm-hmm. Yeah, that's a great question. Thank you. So this was actually the question that we asked when we were looking at the transcriptome profiling, and we were then, really going after this huge selection of marker for the M1 and M2 activation states. So M1 and M2 really would represent tumor suppressive or tumor supportive function, right? So by having a clear picture of whether markers associated with M1 or M2 are present in the infiltrative margin, we were hoping to understand whether potentially those macrophages were either tumor suppressive or tumor supportive. But unfortunately, it's not as easy, at least by looking at the expression. So, and also, there is a literature, I would say at this point is really strong, that really these M1, M2 activation states are not so clear in glioblastoma. So there is a continuum between M1 and M2. So it's very tricky just looking at marker or transcriptome profiling, trying to understand what is the function of these macrophages. So we don't know that yet. Again, what I said is that it's very key to now perform functional assay to direct, really to directly assess the function of these macrophages. Because by using this classification of macrophages, we don't see a clear pattern like, okay, these may be tumor suppressive macrophages, so they are actually beneficial, they're abundant in there, and they may do a good job there, right, to stop the tumor. Or no, actually it's bad to have them in there. That's not clear at this point. But the fact that they are in there and this technique, especially this really resolution that we can achieve by single cell, single nuclear analysis, allow us now really to refine and to understand the heterogeneity of this population. And now really to potentially identify target in those. And if we need to go beyond this really classical classification in M1, M2, we'll do that by looking at the results of the functional assay. In terms of the steroid, I think that's a very interesting perspective that, an interesting comment you're doing. What we try to do in the lab all the time is try to reproduce as much as possible, really what the treatment as well, or anything that patients are given in order then to have cells that can mimic the genetic and the phenotype of the original tumor. So it's actually something that we are considering not only to include the standard of care and to mimic that in the context of the patient-derived cells, but also trying to take that into account and really to interpret the data in light of that.
Okay, thank you.
All right.
You can field questions. I think Dr. Lagari had a question.
Dr. Altaf, you want to speak up?
Yeah, Dr. Piccirillo, an excellent presentation and very, very impressive work you are doing. I was just wondering, in past almost 3 or 4 decades, we have not been able to identify the mechanism behind this nasty disease, which universally kills patients all over the world. And understanding the pathophysiology is of paramount importance. And my first question was, do you think that these macrophages are, it's a kind of continuum of Dr. Chauhan's question, beneficial or harmful for the tumor? So far, we know that these macrophages are a kind of immune response to the tumor cell, which happens to every new or abnormal cell, whether it's a bacteria inside the body or a tumor, and the body reacts to it. So it's a kind of antigen, abnormal antigen to which the body is reacting. And body is trying to curtail the process, but unfortunately in glioblastoma it is not able to. So do you think that this macrophage kind of thing can be utilized, or you can augment it further so that it can curtail the tumor, it can curb the tumor? First question. The second question, do you think that the stem cell, the cancer stem cell, the glioma stem cell or glioblastoma stem cells, which we hypothesize that they are the main culprit behind this disease pathology, have a kind of specific mechanism which recruits all these cells around itself to protect themselves from being killed or making themselves immortal? So these 2 questions, stem cell being protected by macrophages or being augmented by macrophages, or macrophages are trying but not able to kill them?
Yeah, thanks for these 2 questions, very interesting. So to address the first one, again, I think, again, there is a lot of literature, and actually our findings are really in line with what has been shown as well by using a mouse model or all these elegant studies done on so many patients by the GLASS consortium, where they were looking longitudinally at the presence of cells that are not tumor cells, and how those cells, like I mentioned, in the mesenchymal subtype, contribute to this nasty nature of that subtype. In terms of now the presence of these macrophages and their functional identity, especially in the periphery of the tumor, I would say that there are contrasting evidence. So I don't think we are yet in a position to address this question. I think, again, what we need to do is really to look functionally and to understand, again, what are they doing there, whether they are good or bad guys. That's not clear at this point. And also, I would need to say that, up to this point, when you do all these cell type annotation, it's more a prediction. So you really need to go further and integrate and do functional assays to then really understand what these macrophages are doing. And so you need a lot of systems which allow you to study the interaction of tumor cells and macrophages. And you need to recapitulate the microenvironment, which is not always possible. And that's why some people prefer to use mouse model.
Yeah, that's fine.
So I'm sorry, I don't have a clear
answer
to your first question.
Right now?
Regarding your second question, we did a lot regarding cancer stem-like cells in glioblastoma. And actually, by training, really, I entered this field by looking at cancer stem-like cells because I was trained in a lab in Milan, which was a neural stem cell lab. And so there has been a lot of initial findings suggesting the treatment resistance of these cancer stem-like cells and the fact that they are the culprit in really setting the recurrent tumor. But in terms of really the interaction with the non-tumor cells, that's also tricky in the sense that these cancer stem-like cells throughout the years, they have been shown to be extremely heterogeneous. So in work that has been published from my postdoctoral training, but I didn't discuss today, but I mentioned it briefly when I had that slide with the published work summarized. We did that analysis of the cancer stem-like cells phenotype and looking at how they are treatment resistant, for example, comparing those in the subventricular zone with those in the matched tumor mass. And we also look at the phenotype of cancer stem-like cells in the margin. Those are really heterogeneous population. So in order to understand the relationship, for example, with the recruitment of macrophages, which is, I think, what you are alluding to, if I understood correctly your question, we really need to do to take into account the heterogeneity of the cancer stem-like cells and understand how they could do that. Just broadly assuming that what happens in the subventricular zone and the margin is similar to what happened in the most well-characterized area of the glioblastoma patient, which is the tumor mass, I think is not the right way, because by doing all these spatial temporal analysis, genomic and phenotypic analysis, we are really detecting strong differences across this area. So I think we need to go and investigate that aspect further. And like I said, now we are trying to include the analysis of the tumor microenvironment, but we always look at the cancer stem-like cells in each of these areas. So we will do that as well.